Order, order. The committee is now in session. The committee will be hearing evidence today on latest developments in vaccines against Covid-19 and also later on this afternoon on the development of therapeutic treatments. I'm delighted to welcome our first panel of witnesses. They are Professor Zarah Gilbert, who's Professor of Vaccinology at the University of Oxford, and Professor Gilbert leads the Jenner Institute's Vaccine and Emerging Pathogens programme. Professor Sir John Bell, who is the Regius Professor of Medicine at the University of Oxford, and amongst many other distinguished appointments is a former President of the Academy of Medical Sciences. And Kate Bingham, who is the relatively new chair of the UK's relatively new Vaccine Taskforce. Thank you very much all for joining us. Perhaps I can kick off with some questions to Kate Bingham. Kate Bingham, perhaps you could describe the main activities and accomplishments to date, insofar as they are at this early stage of the Vaccines Task Force.
Thank you, Chairman. So I can't tell you too much about accomplishments yet because some of this is commercially confidential. But the goal of the Vaccine Task Force is to protect the UK population through vaccination against COVID as soon as possible, and also to help distribute any successful vaccines internationally. So specifically, what we are doing is to build a diverse portfolio of the most promising vaccines, recognizing that many of them are based on unproven platforms. And what we are doing is to invest both in the manufacturing and the clinical trials before we know whether or not these vaccines will work. We want to maximize the chance of success. And so by having a vaccine available if and when the vaccine is proven to be both safe and effective. But this is an uphill struggle. It's not been done before. We don't know coronavirus well. And in examples like HIV and malaria, we know those diseases well, yet we don't have vaccines against them. So we may never get a vaccine, or we may only get a vaccine that modifies the severity of disease. Our long-term goal, obviously, is to get a sterilizing vaccine, which prevents infection. And so in terms of what we have done, we've worked quite quickly. As you say, this is my 7th week. So we've assembled a private sector expert team, which we've embedded within government. And in addition to that, we've then seconded people from NIHR, which is the National Institute in the UK that runs the clinical studies, and MHRA, which is the regulator which decides whether or not vaccines and drugs are safe and effective. So that means we're getting real-time top advice, which is fab. And in terms of progress, we've had money for manufacturing, and there's a lot more that I can't talk about, but that is all in train. We've been working very closely with the UK's clinical trial network, which John knows all about, and that has been evidenced by this phenomenal result with dexamethasone. And the nimbleness of the small team that we've got has enabled us to move quite quickly. So we are assembling an attractive portfolio. So we're off to a good start, but it is a heroic task.
Thank you. Well, it's great to have that energy behind it. Just in terms of the prospects then for getting a vaccine, how, in your assessment, and you have a lifetime experience in the life science sector, you don't come to it from a position of lack of familiarity. What assessment do you make of the prospect of us having a vaccine eventually? Let's say to start with for COVID. So
I'm relatively optimistic we will find a vaccine that will be able to treat the population. The caveat is the point I made about, is it a full sterilizing vaccine, which means you can't get infected, or is it one that basically just takes the edge off the symptoms so it reduces mortality? And clearly, we would like to get to a sterilizing vaccine so that people are prevented from being infected. But in the near term, we may just have to satisfy ourselves with a vaccine that reduces the severity of the disease. And I'm pretty optimistic we will get that. How quickly it takes before we can get a sterilizing vaccine, I don't have a strong view yet.
And are the different vaccine candidates, in terms of that choice, as it were, between full sterilizing vaccines and ones that help alleviate the symptoms, do the same candidate vaccines do both? It's just a question of how well they work, or are they completely different approaches depending on what they aim to do?
all of them aim to ultimately prevent the infection ultimately. And there are different types of vaccine that work in different ways. And actually, until we run the clinical studies, we don't know. There's nothing fundamentally to say that this one could be a sterilizing vaccine and this one couldn't be. And so we're exploring all the different vaccines. So the adenoviruses, which is what Zarah will talk about, mRNA, which is what we're doing with Imperial and others, and then the more established, and those are the most advanced formats, and they're the ones that we're likely to see the earliest clinical data, but they are the least proven. And then the more proven vaccine formats would be the adjuvanted protein format. So that is what we standardly get. And those are coming a bit further behind, but they are better understood in terms of how the vaccines work.
I see. And is it possible to say which is likely to be available first? Is your implication that a symptom alleviating vaccine is the most likely to be available for deployment, followed by a sterilizing one? Is that the case?
Well, we've got Sarah Gilbert on the line, and she will talk about Oxford. Oxford will highly likely to read out first. And if it's fully sterilizing, fantastic. I'm just conscious that we may not deliver that immediately.
I see. We'll come on to, we've got lots of questions for Professor Gilbert and Sir John as well. But just on this theme, to understand the potential contribution of vaccines, there is a phenomenon, I understand, which is known as disease enhancement, or antibody-induced or vaccine-induced enhancement, where it's possible that a vaccine, obviously inadvertently, and this is why you need to do the testing, can make conditions worse. Can you just say a bit about that and how we can make sure that doesn't happen?
So I'll give an answer, and then I'll let the two experts who know much more about this. So what you're describing is called ADE, antibody enhancement of disease. And this happens when an immune response to a vaccine isn't strong enough to build up a high level of neutralizing antibodies. And so what happens is you may end up with antibodies against the vaccine or against the virus, which enables the virus to actually enter the cells and infect them rather than eliminate them. And so you can mitigate by doing a lot of work to say, are you generating a sufficiently potent immune response to the vaccine, which would then eliminate the likelihood of this happening? And there's been a lot of work done on this. And of course, this is one of the reasons we've got the MHRA embedded in our team, so that we can work every step of the way with the regulator to make sure that anything that we are doing, or the companies with whom we're working, are ensuring against this. But I might ask John, if you want to comment on.
So John, perhaps you might comment on that.
Yeah, so Kate accurately described it, and it is a risk. And there are some vaccines, like the RSV vaccine, that were actually held up 20 years while people tried to sort out the ADE-related issues. I think there's a little bit of encouraging noise, because the convalescent, you might imagine, if that was an issue, you might find enhancement using convalescent sera. And it wasn't really a trial, it was an observational study in the US of convalescent sera in 9,000 patients, I think is just reading out now. And as far as I understand, there's no evidence of enhancement in that population. So that doesn't prove we won't see a problem, but it is encouraging news. And I think Sarah probably also has some data from the preclinical studies.
Professor Gilbert, would you like to amplify the answers that we've heard?
So one way we can look at this is by doing animal studies where we vaccinate them with the same vaccine that we plan to use in humans, and then expose the animals to very large amounts of the virus. And so this is not so much to look at the effectiveness of the vaccine, it's to look at the safety of the vaccine in the face of a very high dose viral infection, and to see if there's any chance that the vaccination prior to the exposure to the virus makes the disease worse. So we've been doing 4 preclinical studies with 3 different collaborators in different parts of the world to look at this. And we had data from the first of these studies before we started our first phase 1 trial. So before any people were vaccinated in our clinical trials, we had evidence from a study in non-human primates, which was made public on bioRxiv, that there was no evidence of disease enhancement after vaccination and then exposure to the virus. Subsequently, we've had data come through from 3 more similar studies in 2 different animal species, and again, no evidence of disease enhancement at all. So we also have a better understanding now what type of vaccine is likely to produce this kind of response. The RSV vaccine in the 1960s was inactivated RSV virus. So you take a large amount of the virus itself, inactivate it with formalin so that it can't cause an infection, and then use that with an alum adjuvant to strengthen the immune response. And that's the type of vaccine that seems to be most associated with causing this type of enhanced disease after exposure to the virus. That was seen in some early development of SARS vaccines, the original SARS, not SARS-CoV-2, again with inactivated virus vaccines with an alum adjuvant. Whereas it hasn't been seen with different types of vaccines, and we now understand better that they're inducing a different type of immune response. So as well as actually doing the studies where we vaccinate and then deliberately infect animals, we can also look at the type of immune response that they're generating and make sure it's the type that we associate with a good response and not with a bad response. And we've also seen that in all of the preclinical studies we've done in the animal testing, and we're seeing in other use of this type of adenovirus vector in the 12 clinical trials we've done before we started the coronavirus trial, we see the right type of immune response. So we're very happy that we're seeing the right sort of immune response that will give protection and not the wrong sort that might cause enhanced disease. Only time will tell if we have any problems caused by vaccination that are very rare events. So if you remember in the H1N1 swine flu pandemic in 2009, one of the vaccines did cause narcolepsy in a very, very small percentage of the people who were vaccinated. And the number was so small that although it's a real effect of vaccination, it's not something you see until you start to do, not just very large trials, but have widespread use of the vaccine. So we can't rule out a very rare side effect of vaccination. Nobody can ever do that, but we can say that we're getting the type of immune response that leads to protection and not to enhancement of disease.
Thank you, that's very clear, and grateful for that. Before I turn to my colleagues, Erin Bell and Mark Logan, just briefly perhaps to ask the witnesses, starting with Kate Bingham, given what you've seen and what you've drawn on in your career, when would you expect a vaccine could be available at the earliest reasonable opportunity?
So again, I'll let Zarah answer for the Oxford one. Outside Oxford, I think it's likely that we'll start seeing vaccines tail end of this year, early next year. So we will have some phase 3 efficacy readouts coming through by the end of the year. And of course, they have to go through the regulatory process to get approved before they would be ready to actually launch. And of course, the issue we've got in the UK is where social distancing has been very effective. And so we are operating in a declining pandemic. Maybe we're seeing a bit of a spike coming up over the last day or so. But trying to demonstrate a clinically effective response, a protective response in a declining pandemic is challenging. So our options, we either expand the size of studies or we go abroad where we can focus the studies in the epicenters. So that is the caution in terms of being able to show a definitive result, is we actually have to have some disease.
Thank you.
But I think the message would be early next year.
But the implication of being early next year is that we might have to face what some people foresee as a winter resurgence without a vaccine. We should be preparing for that, in other words.
I've excluded Oxford. I'll let Zarah talk about her timelines of Oxford.
Professor Gilbert.
Because she's well ahead of the world.
But excluding Oxford then, just on that point, if we take out Oxford, then we ought to be preparing to face the winter without a vaccine before we come on to Oxford.
I think that would be a reasonable assumption, yes.
Okay. Professor Gilbert, how's Oxford in terms of the timeline?
Well, I hope we can improve on those timelines and come to your rescue. obviously, the thing that we have to do is show that the vaccine works. And as Kate said, we can't show that the vaccine works unless we have infections in the group that are receiving the control vaccine, so that we know that people were being exposed and that the vaccine has protected them. And with the transmission dropping so much in the UK, we now have trials going in both Brazil and in South Africa. So we've currently vaccinated a few hundred people in Brazil, but within weeks that should be up to 4,000. And in South Africa, we're aiming for 2,000 people. And those are both areas of high transmission at the moment. It's very dependent on getting the people vaccinated and being able to follow them up and record cases in the control group. And of course, we don't know which group they're in real time. We just know there are a certain number of cases being recorded. And then the statisticians, when there are enough cases, will look at them and determine if the vaccine is working. We're still continuing in the UK as well. We now have 8,000 people vaccinated in the phase 3 trial in the UK, so that's very good going. And apart from the ability to test efficacy if there is any increase in virus transmission, that also gives us the safety database that we need, because we're looking at the vaccine in older adults as well as younger adults. We need safety data, and we need to look at the immune response in people of different ages as well, so that when we do eventually get a result of vaccine efficacy, we can start to make some judgments about whether it's likely to work as well in older people as it will in younger people, and that's really important to know about. So in terms of when we get the efficacy result, I can't give you a precise answer because it just depends on what happens in these different trials that are running. We're now partnered with AstraZeneca, as I'm sure you know, and they will be starting a very large study in the US. In August, they will be vaccinating, and they'll be aiming for 30,000 people, and again in areas where there's high transmission. So we're maximising our opportunity to determine vaccine efficacy, but we have to get that result before we can actually decide that the vaccine is ready to be used. In parallel with that, of course, manufacturing is scaling up, manufacturing is increasing. Every week we're getting improvements in the ability of more companies to manufacture the vaccine, and the aim is by the autumn to have a large amount of vaccine ready to use, and as soon as we have the efficacy results and can go through the emergency use licence process, we would be able to start vaccinating people. We'll come on to the manufacturing in a bit more detail. We're grateful for that. But Kate Bingham said early next year outside your study. You said you could be earlier than that. But can you home in a bit on a kind of time frame? Completely dependent on the results of the efficacy studies. We have to show it works before we can be ready to use it. When we started our phase 1 trial, we were told by the modelers in the UK that if we could get 1,000 people vaccinated by the end of April, we would have a result of vaccine efficacy during May, because the transmission at the time was predicted to be such that we would have been able to get that result. But clearly that didn't happen, because actually transmission was probably higher than the modelers thought it was at that particular time. And so the country went into lockdown, and now transmission has reduced a lot. So these things are very unpredictable, and that's why I can't give you a firm timeline. I can just tell you that we're vaccinating in multiple countries where there is high transmission. So we are increasing the chances of getting the efficacy results early. Okay, thank you. Professor Bell.
One thing I might just add on that is the way that we are working with the NIHR to think about how can we maximise the effect of so that we can enrich for places where the hotspots, where the disease transmission is highest, is where we've got activities in place now that allow us to use the real-time swab data to then send our research nurses to the particular hotspot site so that we can go and enrol in those centres, so that again we can maximise the likelihood that people in the trial will be exposed to disease, and then you'll be able to show whether or not a vaccine actually has a protective effect.
I see. Thank you. So John, and then I'll turn to my colleagues.
Yeah, so my only comment is that this is a world which historically has gone very slowly, the development of vaccines. It normally takes years and years and years and years. And I think to the credit of Sarah's group and the Jenner, they have really changed the way people think about the speed at which these vaccine trials can be done. And I think we'll all remember that when Tony Fauci, who was brought in to help the Americans with their vaccine efforts, in April, he said, don't expect anything till mid-2021. But the reality is that the way that Sarah and Andy Pollard, who is helping to run the clinical trials in Oxford, have approached this has rewritten the book as to how quickly you can do vaccine studies. And we now find many of the other big commercial players are trying to mimic the Oxford approach. And I think that's a great credit to the UK in terms of setting the style in which you can get these things done, but retaining very, very close attention to the safety issues, which is obviously the thing that we need to pay attention to. But many of these companies are now interested, and Kate can probably comment on this, in potentially doing some of their trials in the UK because we actually know how to do it, and we know how to do it because Sarah and Andy have shown us the way.
But do you think we need to be preparing for the winter without a vaccine, or should we be confident that we can have one to help us through the winter?
No, look, this whole epidemic has relied too heavily on assumptions that have turned out not to be true. So my strong advice is be prepared for the worst.
I see. That's very helpful. Erin Bell.
Just one quick statistic for Sarah. I mean, the Oxford study will have likely vaccinated all their efficacy subjects before any of the other vaccines actually start their big efficacy trials. So that just gives you a scale of how far ahead they are versus the other companies.
Thank you. Erin Bell.
Thank you very much, Chair. I'd like to try and just get a bit more of a handle on our current understanding of immunity in general, and obviously it's going to be speculative, but what we might think about the immunity that might be conferred by a coronavirus vaccine. If I could start with Professor Bell, 3 areas in particular seem to be up for debate. The length of any immune response, how long that is likely to be, the effect on different populations, and I think Professor Gilbert already touched on that, and vaccines not always as efficacious with older people. You've covered the disease enhancement already, so the other one would be viral mutations, and would a vaccine still confer some immunity even if the virus itself evolved? So on those 3 issues, do you have any general comments, Professor?
Yeah, so what we know based on other viruses generally, and in particular other coronaviruses, is that the longevity of natural immune responses to the normal endemic coronaviruses doesn't appear to be that long. But in the case of SARS, there was still evidence of antibody-based immunity a couple of years out in infected people, and some evidence in SARS and MERS that cellular immunity continued a great deal longer than that, 6 or 7 years in some cases. So it is possible you get a more long-lived immune response with either natural infection or the vaccine in these cases. I think that how well a vaccine works and how prolonged the immunity it generates lasts is an open question. And again, it's one of those things that we won't know until we've actually been able to measure it. So but I would be optimistic that you might well get certainly a year and maybe a couple of years of pretty safe immune responses. On the mutation issue, this virus does mutate, but it mutates pretty slowly. And it will be interesting to see whether you get a lot of mutation against, for example, neutralizing antibodies. There are programs, as you know, in pharma to generate antibodies that are monoclonal antibodies that are specifically neutralizing. There's a program in 2 or 3 pharma companies to do that. They'll be targeted at particular epitopes or a small set of epitopes, and that will provide a pretty strong selective pressure. And it'll be interesting to see whether the virus is able to avoid those antibodies by mutating to avoid them. It turns out that most of these neutralizing antibodies are just spike, and they don't really always occur in the places you would imagine them. Some of them have rather complex mechanisms of action. So I think that's an open question, but it's an important one. But to a vaccine, as Sarah will describe in a minute, or to other vaccines in this space, they tend to use a much larger protein and will generate a much wider set of antibodies and hopefully a wider set of neutralizing antibodies, which will make them less prone to escape than one sees with a monoclonal antibody, which is specific to a single epitope.
Thank you. Yeah, Professor Gilbert, did you want to add anything on that?
Yes, just to add that before we started the work on this particular coronavirus vaccine, we had been working on a different coronavirus vaccine against Middle East respiratory syndrome, and we had vaccinated people in a clinical trial and taken their serum, which contains the neutralizing antibodies, and tested the serum to see if it would neutralize different isolates of the MERS virus that had accumulated mutations over the time. And in fact, we looked at the most diverse MERS viruses that we could, and we found that the neutralizing antibodies neutralized all of them. So that's a different coronavirus, but MERS also mutates quite slowly. The mutations take a long time to accumulate, and we weren't seeing any escape from the neutralizing antibodies that we were generating by vaccination. On the question of duration of immunity, as Professor Bell has said, it doesn't seem to be very long-lived. Naturally acquired immunity to other human coronavirus infections is relatively short-lived, and we know that people get reinfected sometimes quite quickly, but that doesn't necessarily mean that we'll see the same thing with a vaccine, because the vaccines have a different way of engaging with the immune system. And we followed people in our vaccine studies using this same type of technology to make vaccines for several years, and we still see strong immune responses. So again, it's something we have to test and follow over time. We can't know until we actually have the data, but we're optimistic based on earlier studies that we will see a good duration of immunity for several years at least, and probably better than naturally acquired immunity based on what we know so far. And then in immunity in different populations, different ages, we have previously vaccinated people with this type of vaccine technology against flu and seen very good immune responses in older adults, in people into their 80s. And we are looking at that now for the new coronavirus vaccine, and it will be a question of determining not whether it works at all, but whether the immune response is significantly less in older people. And if it turns out that it is, you might be able to mitigate against that by giving a higher dose or giving an extra dose to try to bring their immune response up to the same level that you see in younger people. But as soon as we have some vaccine efficacy data from any of the vaccine programs, and we have a better understanding of what level of immunity you need to get protection, we can start to tailor the vaccination schedule for older people to make sure that we can achieve it in them, hopefully as well as we do in younger people.
Thank you. Aaron?
Can I just add one question, just one comment to that?
Yes.
Sarah just brought up the point about we could add an extra dose. So the one thing, again, I think is important to understand is these vaccines may not be a single shot. And in many cases, they are already predicted to require 2 doses in order to generate that immunity. So that's another aspect that we have to think about as we're selecting and building up this vaccine portfolio, and then thinking about deployment is what is the likely dosing regime that these vaccines are going to require in order to get that level of protection.
Thank you very much on that. In fact, that speaks to what I wanted to ask as well about, do we need to develop vaccines that might need to be administered in different ways? Whether it's injections or nasal sprays or whatever. Is that something that the programme is looking at, Kate?
Yeah, so our priority focus is on those vaccines that are going into the clinic this year that will generate clinical data possibly this year or early next year. So at the moment, that doesn't include intranasal or oral, although both are absolutely in our sights and things that I think we are continuing to explore for a second wave. But the focus we have is speed, so that we want to basically get hold of the vaccines that are most likely to generate that efficacy data and safety data as soon as possible.
Thank you, that's very clear. And finally, if I could ask the professors if they have any comment on the reports coming out of the Karolinska Institute about the T-cell response being, that seems that it's obviously a small sample and it's pre-peer review and so on, but twice as many people are showing a T-cell response rather than an antibody response. Do you have any comments on that, and does it have any implications for a vaccine programme? Professor Bell first.
So there's actually there are a number of other papers in this space. There's quite a bit of publication going on in this space at the moment, and what seems clear is that you do have cross-reaction from T cells that are activated by the standard endemic coronaviruses, peptides from those viruses, with peptides from the COVID virus. So I think a number of people have made those observations. They're present in quite a significant number of people. So there is probably a background level of T-cell immunity in people before they see the COVID virus. And that may, of course, be relevant the fact that many people get a pretty asymptomatic disease, and the fact that those T cells, there's a concept called T cell senescence, where your T cells get a bit tired once you're beyond the age of 65 and may not be as effective at removing the virus. So that the T cell may explain a number of these different features of the disease. There's quite a bit of work going on in T cells now in the UK at the moment, and we have an opportunity, I think, to see whether immunity to the virus is wholly dependent on antibodies or more likely dependent on antibodies and T cells. And I think Zarah can comment a little bit on her virus and her vaccine and its ability to generate those types of responses.
Thank you. I'll go to Professor Gilbert in a moment, but just to clarify, we've heard this 60% figure thrown about for what herd immunity would look like, or potentially numbers a bit higher than that. But if it's the case that some people have a T cell response without an antibody response, then these prevalence studies would suggest that we would get to a herd immunity position before 60% of the population was showing antibodies. Is that correct?
So it does. This is definitely a confounding factor in the modelers who are trying to think about innate immunity, herd immunity, and are not. I think that's a complicated problem, because I
think we've lost the connection there to Sir John. Perhaps
we'll go to Professor Gilbert for your comments on both my initial question and that follow-up.
Yes, so T-cell immunity is very important in immunity to viruses, and it's something that's often overlooked because it's rather difficult to measure, and it's more complicated to measure, and there are lots of different ways of measuring it that give you different kinds of readouts. What we know is with our vaccine, it's very good at inducing T-cell responses as well as antibody responses, and in the paper coming out of in the paper coming out of the Karolinska Institute, actually a lot of the T-cell responses were to the spike protein, which is the protein that's in our vaccine. And we would expect to get T-cell responses as well as antibody responses to this vaccine. And we've seen that with the MERS coronavirus vaccine. So what it means in terms of vaccine development is if you have something like a protein and adjuvant vaccine, a more traditional style of vaccine that is not very good at inducing T-cell responses but does induce antibodies and potentially neutralizing antibodies, you may need to have a much higher level of antibody induction than you do with the viral vector vaccines and the RNA vaccines that induce T-cell responses as well. So because you've got two components that are contributing to immunity, so you may not need such a high level of antibodies to get full protection if you've also got a strong T-cell response which is adding to that protection. And in terms of the seroprevalence may possibly underestimating naturally or already acquired immunity to this virus. I think that's something that we really need to keep an eye on, because there are certainly evidence that people have been infected with the SARS-CoV-2, have not developed antibodies, but do have a T-cell response, and that will be likely to protect them against another infection. It's not massively more than the people who have antibodies, and it also depends on which assay you're using to measure the antibody responses, because if the assay is not very sensitive, then you won't see an antibody response. If you use a different assay, you may see an antibody response. So I think we have to keep an open mind about whether there are large numbers of people who have protective T cells in the complete absence of antibodies.
Thank you.
Thank you very much indeed. So I'm going to go to Mark Logan, then Andrew Griffith and Dawn Butler. Mark.
Yeah. Thank you, Chair. And just following up from the questions Erin had around immune response, this is a bit of a layman's question, but if we did have a vaccination in place by the winter, the fact that we every year nationally roll out flu injections for people in advance of the winter, does that have an impact on people's immune response system, or those who have compromised immune systems? Or should we be thinking of rolling out the flu vaccination much earlier this time round?
Shall I start off with that?
Yes, please, Kate Bingham.
So it's the JCVI, so the Joint Committee of Vaccine and Immunisation, which will advise the government on what is the right way to think about who should be vaccinated and when. So that's the starting point is we have expert people that will advise, and they've already issued their first advice on the likely vaccine prioritisation. The second point to make is that none of the trials at the moment are being co-administered with a flu vaccine. So it is not likely initially that people coming for flu vaccines will be able to have a COVID vaccine at the same time. So again, that means there will be a separate deployment process to think about how you vaccinate those different subpopulations. But the advice that's come from JCVI is it's likely that the expanded flu cohort, so those people who have been recommended to have annual flu vaccinations, are likely to be the same people initially who will be recommended for COVID vaccines.
Okay. Mark.
Greg, can I just come in on this? Because I think it's a pretty important question. I think one of the things that clinical staff in hospitals worry about is if we do have a significant flu season, then we are going to have a bit of a clinical problem if we've also got COVID running alongside, because you'll get people with severe pneumonias arriving with fever and all the usual things, and it'll be pandemonium in the A&E department. So what I'm rather hoping is that we first of all, we ideally expand the number of people getting flu vaccines so we get better coverage. And secondly, we push quite hard to make sure people are compliant and participate in the flu vaccine programs. The uptake is lamentably small in some sectors, and I think we're not messing around anymore. This could be really serious if people don't get their vaccines.
And so, John, to do that, do we need to emphasise Mark's point? Do we need to start earlier? Do we need to be doing it in the early autumn rather than the later autumn?
Yeah, some of this has got to do with the availability of the vaccines. As you know, they take some time to make, and they have to be made every year. And also being able to predict exactly the subtype of flu that's going to be used is not an easy game. I see there's a new H1N1 just popped up in China. So I think when they're ready, we should start when they're ready and get going, would be my view. But I think more importantly, it's surprising how many people who are eligible for flu vaccines don't get them. And I think we have to really enforce the idea, well, not enforce, but encourage people to participate as widely as possible.
So John, can I just ask then, because it sounds like being prepared for the first wave will not be the same as being prepared for the second wave. Is that what you're kind of implying? Because we'll have different conditions.
Yeah, it's exactly what I'm implying. I think, this, whatever happens is likely to happen through the autumn, getting into the winter, and we'll have a whole new set of other respiratory viruses floating around. And if we happen to have a bad flu season, it will cause lots of trouble, and there will be a real challenge, doing diagnostics, separating the flu from the COVID patients, doing all that kind of stuff. So we need to be on the front foot, is what I'm saying, and be prepared for that come the autumn and the winter.
Thank you. Mark?
Thank you. And then just a question for Ms. Bingham is around what potential stalling points in the vaccine development process has the task force identified, and what action has been taken to remedy these?
Well, there's all aspects of vaccine development. So it's you've got the manufacturing supply chains, which is can you actually make the vaccines? And so what we've done there is to put in place 2 vaccine supply chains, one for adenoviral vaccines and one for mRNA vaccines, so that, and those are currently supporting Oxford and Imperial, but those are generic supply chains that should those 2 vaccines fall over, we'd then be able to use those manufacturing capabilities for alternative vaccines. We have plans in place I can't talk about today to expand capability and capacity in the UK, but what is public is VMIC. So VMIC is the Vaccine Manufacturing and Innovation Centre, which is just being opened now in Harwell. And that basically, we've now expanded both the capability of the different kinds of vaccines it can make, as well as the capacity. So the capacity of VMIC, when it comes on stream, is to, at surge capacity, make 70 million doses of vaccine in 4 months. So we're basically shoring up the supply chain so that we can actually manufacture vaccines as and when they look promising and we want to bring them into the portfolio. The second thing is around the clinical trials. So basically, as we've talked a little bit about, how do you actually prove that a vaccine is both safe and efficient? And so what we need to do is to be able to ramp up the clinical trials very quickly. As soon as a company is ready to go into those efficacy studies, we need to be able to turn those studies on in a dime. And so in addition to using the real-time transmission data where we can send research teams out around the country to Leicester or where the hotspots are, we've also assembled a group of pre-existing clinical trial cohorts. So these are people who have already signed up to say they're willing to be approached for clinical trials. And we're focusing those cohorts on ones which we think are relevant for the coronavirus vaccine. So that includes cohorts which have ethnic minorities. So we're working with a genes and health cohort, which combines people from East London, managed by Barts, as well as Bradford. So that's 47,000 British, Bangladeshi and British Pakistani volunteers that we've already got that we will approach in due course for vaccine trials. And then we've got other cohorts which are pre-assembled, dealing with elderly people, so that again, another likely high priority population for vaccination, so we can include those in clinical trials. So the two key things that we've really focused on is how can we be sure we can make enough vaccine quickly so that we've got sufficient supplies for the UK, and how can we be sure that we can demonstrate that those vaccines are safe and effective and do it as quickly as you possibly can. I've never seen trials get started as quickly. I mean, an example, obviously, as I said, is the recovery study. Is phenomenal. It's just rewritten the rule book about how you get clinical trials up and running and controlled and end up with a very definitive result at the end of it. And this is what we need to get out of it in the UK, is to show that we can be the place in the world to do these sorts of highly high quality and nimble clinical trials with the really definitive data coming out.
Thank you. Mark?
Thank you. Yes, and just to go back and link to that, to something Professor Gilbert was saying earlier about how the transmission rate decreased much earlier.
We've lost the sound, Mark. We'll try and come back to you.
Going to be the case with the planning. So I was wondering, should trials of UK developed vaccines?
Mark, we've lost the sound on you.
Is this okay?
No, we'll try and come back when we get a better connection.
I can hear you again.
I'm going to come to Andrew Griffith, if that's okay, and then we'll try and come back to you later. Andrew Griffith, then Dawn Butler, then Katherine Fletcher.
Thank you, Chair, and thank you for everything you're doing, Kate, Sir John Bell and Sarah Gilbert. Thank you. It's a really good, positive, upbeat session, if I may say so. I just want to talk briefly about funding. We had evidence from Phil Duffy back at the beginning of June, who talked about, I think it's about 114 million of funding.
And Mr. Duffy is from the Treasury. Perhaps the witnesses might need to know. Yeah.
Always good to know exactly the name of where your money's coming from. I just wondered if you could give us an update. Has all of that money flown to your programmes? Is there more money that you need? But perhaps you could give us an update, starting with Kate.
Yeah, so I absolutely know who Phil Duffy is, and he's excellent and has been highly cooperative and helpful. I'm afraid 140 is not going to be sufficient. We have a funding plan submitted with the Treasury now. And a full business case. So I can't give you the details of that, but suffice it to say that the teams are working very well. It's coming up for approval or not, and we haven't had pushback on anything that we have requested to date.
Right. To that point, the Vaccines Taskforce originally said that you were going to develop a funding and operational plan for everything we've talked about today for procurement and delivery of vaccines. Has that plan been submitted now? And would you be willing to publish that when you do submit it?
Well, the plan has gone through the BAYS process and is being considered at the programme board coming up. I don't know the political aspects of whether or not it gets published or not. I just don't know the answer. We'll have to come back to you on it.
It exists. It's just... You've pushed that up into the system now.
We are very clear on what are the the funding costs, the milestones, the key value inflection points, at the point at which we say, yes, this is working, or no, this is not working. So one of the things, I'm a venture capitalist, one of the things I'm very, very sensitive to is not spending money if you don't need to. And if it's very clear that something's not going to work, then you turn the lights off and you move on. So you focus your funding and your resources onto those candidates that are most likely to work. So yes, we've got a very clear plan of what we're doing.
So to that point, where would you spend now if the magic money fairy comes along? Where would you think we would get? And I know John's always full of ideas. I might direct this to Sir John. But where would be the best place to put incremental funding should we want to try and move further or faster?
Well, I think that the game here is to place lots of bets, because we're not. Look, hopefully Zarah gets a tailwind and she gets across the finish line. That would be terrific. But I don't think we can assume that's the case. So we want to be prepared and ideally be ready to manufacture at risk for a number of other vaccines. And as you know, there's 170 vaccines on the list, so we don't want to do them all. But I think to give Kate the elbow room to do a significant number will be important. And because our vaccine manufacturing infrastructure in the UK was allowed to drift and was pretty lamentable, to be honest, at the beginning of this activity, we may need to think about putting money into infrastructure to allow us to do the things we need to do between now and next summer. And I would strongly encourage that, because we don't want to be left relying on importing vaccine from the US, because I promise you will not get it till a long way down the line. So I think we do need to be self-sufficient in this space.
But just to be clear, that is what our plan includes.
I was going to say, yeah.
Our plan has funding for the clinical, well, first of all, it has funding for a portfolio of vaccines. Second of all, it has funding to run the clinical trials for those vaccines. And then it has funding to do the bulk manufacture and the fill and finish manufacture of those, both in the short term using CDMOs, which are contract development organizations, as well as building permanent manufacturing capability and capacity.
And orders of magnitude, I didn't think you would say that 114 million was enough, but orders of magnitude.
We haven't disclosed it.
$1 billion? 2 billion? I mean,
I'll give you. It's more than $1 billion. Yes.
Yeah.
But until we've got the sign off, I think I shouldn't say where we are.
No, I fully understand. Sorry, I wasn't trying to overly press you, but
the material sums, because we've got to do this properly. We can't do a half committed plan here. We're doing it properly.
Yeah. So the status of that, the plan exists. The plan you've put together does what you think we need to do as a country, not just for the short term, but to John's point, to create long-term capability in this space, potentially to create future economic opportunities for the country as well. And that is going through the process at the moment.
Yes, and skills, because of course, as we build up the advanced manufacturing capability, we also need to include the people who are actually going to be making it and doing it. So that actually something that we're obviously investing in as well is how do we make sure that we can build those jobs and build the skills workforce to actually deal with the advanced manufacturing.
Sounds like the sort of thing you want to wrap into a New Deal.
So Andrew, there's a really important point here, and that is that to be honest, we were pretty complacent about our abilities to manufacture these kind of health security type products. And I think you probably know, but the truth is we have very little onshore antibody manufacturing capabilities. So if neutralizing antibodies are the answer, we're going to be stuck with that as well. But I have made the argument, and fortunately when Greg was the Secretary of State in Bayes, he did listen, and the VMIC was entirely the result of an effort to build out some activity in the vaccine manufacturing space. I think what people need to remember is this is not a one-off, and we're going to wait 100 years for another one. Since 2000, we've had 8 close calls, which should have made us wake up. We've had 3 flu epidemics or near flu epidemics, 2 SARS episodes, 1 MERS episode, Zika and Ebola. And any one of those could have blown up to be exactly what we've got now. So This, I think, now needs to become part of our national health security infrastructure. It's really important.
Thank you. So John
we
is
need
being...
to make sure that we come out of this stronger as a result of the pandemic than we went in. So that is a very clear mandate for us to deliver. Absolutely.
So John's role in this should be recorded. He was self-effacing, but he proposed in the Life Science and Industrial Strategy, the Vaccine Manufacturing Innovation Centre. I was pleased to support it, but it was John's idea. Can we go to Dawn Butler, if that's okay, Andrew?
Yeah, that's fine. I just didn't, unless there was anything Sarah wanted to say, I didn't want to discriminate when the money's being talked about, but I'm done.
Just to say very quickly, this is something that we need to do more of, but it's also something we're very good at. And we really shouldn't just make one vaccine and stop and then think that it's all behind us. As Sir John says, we need to do more of this, but we can do more of this. We need support to continue and do it faster next time.
Thank you. Thank you, Professor Gilbert. Dawn Butler and then Katherine Fletcher.
Thank you very much, Chair, and thank you all for all the work that you're doing. Can I just pick up on something that was said earlier in regards to 2 vaccinations that will be needed? Is that 2 vaccinations straight away, or does there need to be some time lapse between vaccinations?
So it's likely the vaccines that we're looking at that have 2 different vaccinations are between 21 days and 28 days apart.
Right, so you would need to see the person you vaccinated twice. Right, okay, that's interesting. And Kate, you talked earlier about how fast we've been able to develop them and move quite speedily as opposed to what normally would be the case. What would have been the normal hurdles that would have slowed down this process that no longer is there?
So both the two strands. One is the manufacturing. So if you think about, if you take Sarah's vaccine as an example, growing up a modified virus in cells, you start off in a test tube and then you slowly take it in a stepwise way and you scale it up into thousands of litres. And what we're doing now is doing that incredibly quickly rather than making sure that each step is completely validated and every single aspect is checked in terms of optimizing the processes and the reagents and the conditions. We're basically accelerating that process. So there will be bumps as the way because we simply can't predict how the cells will perform in different quantities effectively. So that's one aspect, which is we're doing it much faster, but there will be some unknowns and things will get in the way. And then the other aspect is the clinical trials, so that we are absolutely compressing the clinical trial process. And so again, that's why having the MHRA embedded with us, and MHRA are, I think, now universally known to be an incredibly flexible and thoughtful regulator. So we're hearing very good comments from the different companies that we are talking to around the world who are now preferentially wanting to come to the UK because they know that the MHRA is willing to talk to them at a very early stage and look at the data and the documents before they've even sort of got beyond a draft stage so that they can give input along the way and therefore be able to turn it around quickly. So those are the 2 aspects that are being compressed. Now, inevitably, we will have some unknowns and some bumps. So don't think that it's all going to work perfectly. But I think, as John said, we will come out of this stronger because I think the streamlined ways of working that we've had to develop as part of COVID absolutely sets the pace for how we should be doing things in the future.
Thank you. Dawn?
Professor Bell, what do you think those bumps will look like? Do you have any idea in your modelling what you think those bumps will look like?
Well, there are a whole set of potential things that may get in the way. One of the things is that the vaccines may not show the efficacy that we need, so that's an issue that'll push the issue further down the road. There is a massive issue about manufacturing at scale. Sometimes the scale up doesn't work. Getting material in place is going to be complicated. There are also a set of wider bumps that relate to not just the UK, but the world getting access to this vaccine. So whatever vaccine comes through, I think we all hope that it will be made available globally as quickly as possible. That is a mammoth exercise, and certainly the partner, Sarah's partner in the form of AstraZeneca, have been very focused on trying to make sure there's supply for the developing world, for example, low middle income countries that may not be able to produce it themselves. Because this is a situation where we're not safe until we're all safe. So there's no point in saying, well, we'll just vaccinate the UK and everybody else can worry about it themselves. There's actually a corporate responsibility to make sure that everybody gets vaccinated as soon as possible.
Thank you. One last question, Dawn?
Can I do two more, Chair? I mean, just on a follow-up to that, or lastly, I was going to ask about international working, and are there any countries that are ahead of us in regards to the development of the vaccine? But I also wanted to ask Professor Gilbert just about the trials and the selection process for the trials and people signing up for the trials. How exactly is that decided, and are people signing on the dotted line, and are they fully informed of, you know, that there could be complications? How's that actually working?
Thank you.
Okay, so no country is further ahead with us in development. In terms of recruitment to the trials, we publicise the trials on the website. People are invited to register and receive information. They're sent an information package. They have to be within a particular age range for different components of the trials, and there are some things that would rule them out, such as, you know, severe disease, severe pre-existing disease, for example. We then invite people to come for what we call a screening visit, where they get the opportunity to discuss the trial, the information they've received. They see a video presentation to explain everything to them in that way, and then they have a one-to-one session with a doctor where they can ask questions. They have blood taken to check their health before they enter the trial, and then they have a consent form which they can choose to sign to say that they've understood all of this information and that they want to be part of the trial. So then there will be a gap between the screening visit, when they have all of the information, and the vaccination visit, when they then return. So they have time to read everything, discuss it with family if they want to, think about it, and then they're invited back for a vaccination visit. And before they're vaccinated, obviously, they would need to sign the consent form. So it's not a rushed process, and they have plenty of opportunity to receive information which is all approved by the ethical committee, and then ask questions about it before they decide if they want to go ahead.
Thank you, Professor
And is it all done
Just
in different languages? Sorry, Chair. Just wondering if it's all done in different languages as well.
In the UK, it's not done in different languages. Obviously in different countries, then all of the public-facing information has to be translated into the local language, and depending on what country it is and how many local languages there will be multiple language documents provided. So when we do trials in the Gambia, for example, or in Kenya, there may be more than one language that the documents need to be in.
Thank you.
Just
in a sentence, I wonder whether Kate Bingham or Sir John Bell could say whether any countries ahead of us in this race to develop and manufacture vaccines.
Can I just come in?
Sir John and then Kate Bingham. Sir John?
Well, Oxford is the head of the world in terms of it's the most advanced vaccine anywhere in the world. And
I'm sure the Regius Professor of Medicine at Oxford is going to agree with that.
Yeah, but can I just add that I know everybody thinks that we've had a bumpy pandemic. This is stuff that the UK does unbelievably well, much better than anybody else. And it's not just Oxford. It's the vaccine Taskforce, it's the clinical trials. I mean, it's amazing, and we should be very proud of that.
Great. Thank you. We are. Let's turn to Katherine Fletcher now, please.
Thank you, Chairman. Given you've given such a rousing, I have been trying to explain the unbelievable efforts that have been going on in this space to friends that perhaps aren't close to the detail. And I've been describing this vaccine effort, should it come off, as something not far off going into a shed and coming out with a jet engine. I wonder where, yeah, I can see nodding. It's, you know, it's pipe cleaner stuff. And I wanted to, forgive me, I wanted to just take this opportunity to say a big thank you for the women-led science that's going on here. I was trying to explain to my niece when we heard private evidence from Professor Gilbert way back when, and it all went in, and then she said, about 3 weeks ago, she said, how is the very clever lady with the camels getting on? So, but no, in all seriousness, your leadership is an inspiration to a generation, and I just want to put that on record. Thank you. Now, I was going to talk about a UK vaccination strategy, but we've actually started to cover it off slightly. So if I understand correctly, we have a strategy if this vaccine candidate's brilliant, and we have a slightly different strategy if that vaccine candidate's brilliant, but the nuances require the clinical evidence. But we've got manufacturing capability for a range of different vaccines should they be required. That's correct, isn't it? Are there any concerns that the committee needs to be aware of about a strategy for UK vaccination? Although, Professor Sir John, I know you're correct about the need to make sure that the world is safe. So if I could perhaps start with you, sir. Is there anything we need to worry about in a UK vaccination strategy, or anything the committee needs to focus in on?
I think Kate and her team at the Vaccine Taskforce has covered off almost all the key risks that we had. We had, to be clear, I was on the task force before Kate ever joined it, and the list of risks was very long. But as far as I can see, they've looked after most of the things that it would make me anxious.
Catherine?
I think the one key risk is the fact that some of the vaccine candidates we're looking at, specifically the mRNA vaccine candidates, require a very challenging cold chain. So that some of these vaccines have to be kept at -80 degrees. And they're all obviously working very hard to say, well, what level of degradation do they get if you warm them up to -40, -20, -2? And that, I think, is a challenge. It's not a challenge specific to the UK, but it is a massive deployment challenge, and it will make it much harder to distribute those vaccines internationally until we have a solution for how can we stabilize them at temperatures that aren't quite so heroic.
Yeah,
Catherine?
Understood. Just one final one from me, Chair. Do you have any concerns about the behavioural side of this in terms of adoption of the vaccine from it's not an area that's without its controversies in childhood vaccination, for example. Do you have anything that suggests it's something we need to think about? Sorry, I haven't directed it to anyone. Big Kate.
There's a very active anti-vaccination lobby, and we need to recognise that is out there. And John talked earlier about saying that not everyone is necessarily keen to get vaccinated. So I think what's important, again, is that we work within the regulatory framework and make sure that what ultimately comes out of this process, if anything comes out, is safe and is approved by regulators. And yes, I absolutely think we need to work on the communications and work on being clear about what these vaccines can and can't do.
I'll repeat my thanks to all three of you.
Thank you, Catherine. And finally, just a couple of questions from me and my colleague Graham Stringer to Sir John. Sir John, you're a professor of medicine, and you have taken a very authoritative view through the whole pandemic, I know, advising very wisely bodies and governments in this. There was a study in Nature published yesterday that found in one of the hotspots in Italy, over 42% of people with COVID infections were asymptomatic, showed no symptoms at all. Does one draw from that the next phase of the response has to be routine testing of people who are not displaying symptoms?
Yeah, so the data from the ONS survey, which I helped to set up, as you know, here, which is doing a representative sample around the UK, comes up with a number which is higher than 40%. In the ONS survey, it looks like it's 70%. And because people are revisited every week, we can know not only do they not have symptoms today, but they don't have it next week or the week after. So I think that number is probably pretty robust. So this is largely an asymptomatic disease, which basically means you have to be careful about a track and trace and isolate strategy that relies on symptoms, because you'll miss 70% of the people. So my view, which I have certainly expressed before and continue to do so, is that we do need much more extensive testing across the community, not just the big labs, which have been terrifically successful, as you know, but also distributed testing now at sort of point of care level. And I think here we need new technology to help us. but I've seen in the last week or two a couple of examples of tests that might well be of the standard we need that can produce a result in a few minutes. And that would be transformative, because we could all test ourselves regularly and test our kids after they've been off to the rave and all that stuff. So I think that's sort of where we need to get to.
And would it be fair to say that the priority for those tests, those regular tests of the asymptomatic, should be in NHS settings and care homes?
Well, in a curious way, NHS should be able to deal with regular testing because they have the big PCR capability within the lab. So I think they can saturate tests as much as they want. They got off to a slow start, as you know, and I'm not entirely sure why that was, but they really should have been testing from the get-go, all their staff. Care homes the same, got off to a slow start. But I obviously the more widespread the testing innovation becomes, the easier it'll be for care homes, for example, to test regularly.
And now we're told, plenty of capacity in testing. Can you think of any reason why you wouldn't test regularly and routinely in hospitals and care homes?
No, I can't think of any reason. I think, managing, if we want to manage this infection, as we know from the Italian experience and to some extent the UK experience, when it gets away on you, it often gets away on you in hospitals. So saturation testing in hospitals, I think, should be an absolute requirement. And I'm not talking about antibody testing. I'm talking about real PCR virus testing, because that's what tells you who's got what.
Thank you. That's very clear. Graham Stringer.
If I can take you back, Professor Bell, to something you said before. You painted a pretty horrific picture of next winter if there's a major flu epidemic. I've not seen any statistics, I assume they exist somewhere, of the last flu season from the winter that's gone by. Do we know what impact the last flu season had on this epidemic? Has there been any contamination of the statistics from that? And can we learn from that about any potential flu epidemic interacting with the continuing COVID epidemic next winter?
So the answer is last winter was not a bad winter for flu, which was really fortunate, because if it had had a tail into February and March with bad flu, we would have had a real problem. So we were lucky. It also doesn't appear that Australia at the moment are having a particular issue with flu. And usually the two goes hand in hand. You get a bad winter for them, and then you get a bad winter for us. But there are these reports coming out of China about a new flu strain, which is a swine flu strain, which always worry you, because many of these strains do emerge out of China. And if that ever got going over the course of the next 4 or 5 months, we would have a big problem.
Thank you. You referred to the NHS getting off to a slow start, which is, I think, a bit of a euphemism. Listening to you as witnesses today, and I don't know if this is the 11th or 12th session we've had, you can't, as a member of this committee, think that anything but that the UK is a scientific superpower, because we've seen absolutely first-rate scientists before us. But when it has come to testing and tracing, we seem to have been second or third rate, potentially over-centralised. Would you agree with that? And as a close observer of events, can you give any reasons why that might be?
Sir John, I assume.
Well, I think there's a general, can I just paint a general picture? And that is, I don't think we had particularly competent or extensive planning for any pandemic before this, to be honest. The plans for pandemics were not very well developed. People occasionally talked about flu, but very little was done. I think we weren't really ready for a pandemic in the UK. And I think that's something that we need to pay very close attention to, as I said before, going forward. I think where we have shone, and I think you've identified a couple of areas where we have shone, the ability to do clinical trials at scale with remarkable ability, the ability to produce vaccines which are well ahead of the rest of the world. they didn't come from a pandemic planning strategy held by PHE. They came from strong academic science and have been allowed to flourish in this environment. And without them, we'd be in real trouble. So I think we have to go back and think about how our healthcare system and our public health system thinks much more strategically about how we get ready for pandemics, rather than try to react after they've arrived on the doorstep, because I think that's where the problem was.
Final one, Graham?
One more, if I may. Professor Bell, I think that was very diplomatic and, in one sense, clear answer. There's been a lot of press comparisons, and in the academic press as well, between how this country has dealt with the epidemic and how other countries around the world have dealt with the epidemic. There have been difficult comparisons with what has happened with the epidemic in Japan and South Korea and Taiwan. Do you think the differences are down primarily to their ability to test and track very quickly, or are there other factors at work that would help us understand the very different infection rates in those countries?
Yeah, so I'm not persuaded that the infection rate or death rates in those countries can be attributed to the testing regimes. They have often had better testing regimes, but South Korea hasn't necessarily done that much testing. Where South Korea has been rather good is contact tracing and identifying people who have been in contact with others who've been infected. And similarly, the German experience, which is, I think, really the exceptional experience, I don't think you can attribute that to the testing in Germany either. The testing in Germany was very distributed. A lot of it was done in primary care. It was not clear to me that the data was centralized or handled in a way that would allow them to make strategic decisions on the back of it. I think there have been some countries that have been lucky and others that have been unlucky. That would be my view.
If you don't mind, if I could just, that doesn't sound like a very scientific answer, if you don't mind me saying so.
No, it doesn't, but I can't produce a scientific explanation. And as you can see, now that we get to the second wave, Germany's got its own problems. So it's not entirely clear why they avoided the first wave. But it's not clear that, the testing itself was going to would reduce the death rate to such an extent. Because testing people, unless you follow up with really extensive contact tracing, will not reduce the death rate at all, because we can't alter the course of this disease, at least until we discovered dexamethasone 2 weeks ago. So.
And you don't have a hunch what the difference might be?
I've got a lot of hunches, but I think some of this may be due to immunity, but I don't want to go any further because I have no data.
Thank you.
Thank you. I'm sure we'll have a chance to talk about these things again. Can I reiterate the remarks of all our colleagues and how much we appreciate your expertise being deployed throughout this pandemic in the public interest. You have a very important task ahead of you. We wish you well in it, and we're very grateful for your very clear evidence today. Thank you very much indeed. Order. We'll suspend the hearing for just a few seconds while we arrange the next witnesses. Thank you. Order. The committee is back in session. I'm delighted to welcome for our second panel discussing some of the therapeutic innovations that are under development. Professor Jonathan Van-Tam. Professor Van-Tam is a famous face now throughout this pandemic. He is the Deputy Chief Medical Officer and is also Professor of Health Protection at the University of Nottingham. Thank you very much indeed for joining us today. And Dr. Jonathan Sheffield, who is the Covid-19 Research Operations Director at the National Institute for Health Research. Thank you, both witnesses, for coming. Perhaps I could start with Professor Van-Tam and just ask you to summarise the purpose and what you regard as the achievements to date, recognising that it's still early, of the UK Therapeutics Taskforce.
Thank you, Chair. So I think I could summarise the purpose of the Therapeutics Taskforce very easily, and to say that we started this pandemic with no known effective treatments, and the mission of the Therapeutics Taskforce is to get as fast as possible to discoveries, and that doesn't necessarily mean new drugs, but discoveries of medicines which are safe and at least partially effective, that are accessible to UK patients primarily, but of course then with one eye further afield on their accessibility across the world. So I think it's about discovery, about safe and effective, and about UK patient access and incorporation into UK clinical practice as rapidly as possible.
Thank you. That's very clear and succinct. And I think it would be fair to say that dexamethasone would conform to that. Perhaps say a bit about that.
Yeah. So the really big achievements so far of the Therapeutics Taskforce are, first of all, to identify the very first medicine in the world that is proven to reduce mortality from Covid-19. Now, I understand that there are also discoveries around remdesivir, an antiviral drug, for example, but the data on mortality are not clear in that particular space. But dexamethasone, I think the data are very clear from the UK effort that it reduces mortality. On top of that, dexamethasone in comparative terms is an extremely cheap and extremely widely available drug. So that is extremely solid and important news for UK patients and for global public health. The additional discoveries already are that a couple of medicines widely touted as being important for the treatment of Covid-19, and you'll have seen yourselves the headlines around the world, hydroxychloroquine, the anti-malarial drug, and Kaletra, which is a combination of two HIV drugs, both of which have been claimed to probably be of benefit to patients. I think our efforts in the UK through the Therapeutics Taskforce have conclusively now arrived at a conclusion that they are not beneficial. And I think that's the really important piece here about the clinical trials that we've done. They have established for and against certain drugs.
I see. And very helpful and important information. Can you say why it is that the task force that we have and that you have lead in this country has been able to do this ahead of the world? What do we do? What do you do differently?
Yes. So I'll say first of all that I don't agree with the previous speaker that we were not well prepared and one of the best prepared countries on the planet for the advent of an influenza pandemic. I personally think we were, and that's probably a discussion for another day. Mm-hmm. But why it's contextually important to say that to you now is that after the 2009 swine flu pandemic, as it was known, there was a very important reflection exercise in government. I wasn't part of government at that time, but I was one of its advisors. And the reflection was that in 2009, we had to get an awful lot of research going very fast, really from a blank sheet of paper. And after that, the mentality changed. And there are a long list of studies for pandemic influenza that were prepared, that were rehearsed, funded and then mothballed, ready for an influenza pandemic. Now, we didn't get an influenza pandemic, we got a coronavirus pandemic. But that thinking about, first of all, getting research moving much, much faster, and indeed repurposing some of the studies that had been prepared for pandemic flu to mobilize them for a new purpose, to the coronavirus pandemic, that happened. So the first part of the answer, I think, and the most important part, is that actually the culture had changed since 2009 to now, that we were ready to move very fast indeed to start research. And that was one of the earliest conversations I had with the Chief Medical Officer after it became clear that this was going to be a pandemic, about how do we get moving on this.
I see. And so in terms of the example of dexamethasone, the early progress you were able to make with that, you think draws on that experience since 2009?
I think it draws on that mentality.
I see.
Yes, absolutely. And that culture that we had to get things moving very fast indeed. And as you know, it was a matter of the dexamethasone discovery was part of the now celebrated recovery platform trial in the UK. And it was really a matter of, I'll ask Dr. Sheffield to give absolute chapter and verse, but it was a matter of a couple of weeks from concept through to first patient being recruited to that study. Absolutely. It has been the most dramatic success in terms of activation and rapid recruitment that I've ever seen, that I think anyone has ever seen internationally.
Thank you. Thank you. Well, we'll come into some more detailed questions on this. Now clearly, we have an enormous appetite to make these discoveries and to establish the safety and the effectiveness of them as soon as possible. But in the case of dexamethasone, the preprint that was published on the 22nd of June did carry the standard warning that it was a preprint and should not be used to guide clinical practice. So how did you form the conclusion that, notwithstanding that early stage in terms of the publication cycle, it was nevertheless safe to be deployed?
Yes. Well, we were informed about the dexamethasone result. We had a discussion with the principal investigator on the I think it was the 14th of June, and we were very clear it was very clear from them that they intended to independently announce the result on the 16th of June. We understood the recovery trial from the word go. We'd seen the protocols clearly. We'd approved them because it was a UK government-funded trial at the end of the day. We understood the absolutely enormous patient numbers in the treatment arms. And we understood from that perspective that the size of the result they'd got, the statistical significance of their findings, meant that the probability that this was a result that had arisen by chance was so incredibly low. And we had we still have a very high confidence in the absolute quality of the recovery team. So much so that we felt actually it just wasn't the right thing to do, to wait more than another day to allow UK patients to begin to access that.
Thank you.
And I think it was a collective system judgment and a collective system taking of risk that we were prepared to operationalise that 2 days later with a letter to all clinicians in the NHS advising them with the caveats that this was pre-peer-reviewed research, as it still is, unless things have changed sometime today, that we should go ahead immediately. And I'm glad about that. I make no apologies for that at all, for allowing UK patients access as quickly as possible. And in the same tone, I make no apology for the fact that we took a decision many weeks before that to put the UK in the best possible position, that if one of these medicines was going to prove to be effective and important, then we had to have supplies ready to use, and we do.
Absolutely. But one of the things that we're keen to do is to document and to analyse and commend, if necessary, innovations that take place, and speed, we know, is of the essence here. When you referred, Professor Van-Tam, to we in this, what was the approving body Who's the we in the decision to approve the treatment?
Right. Okay. So that decision was taken at the highest level by the Chief Medical Officer. It was entirely a clinical decision, and it was informed by an expert senior clinical group, but ultimately the decision is with the Chief Medical Officer.
And it comes from the clinical side of his work rather than the SAGE side, as it were?
Yes, it does. It comes from the clinical side. Does SAGE really would concern itself primarily with the science.
Absolutely. And just one final thing before I turn to my colleagues. Remdesivir has been in the news today, and we understand that it's likely to be the first treatment approved at the European level. Have you made an assessment of its prospects over the last few weeks?
We've been keeping a very close eye on Remdesivir indeed, and I do understand that the CHMP, the European Approval Committee, has made a provisional recommendation in favour of Remdesivir. That will take now a period of time, usually a matter of a few weeks. It's mainly procedural before there would be a European licence for Remdesivir. The decision has been made on the basis of the US clinical trial data, which shows that patients who are hospitalised with moderate severity with COVID-19, their length of stay in hospital is reduced. There is a suggestion, and I think I'd call it no more than that in the data, that mortality may also be reduced, but that's not a clear or statistically significant signal. The size of that trial is completely dwarfed by the kind of trial size and comparison size that recovery can make. And I'm cautious at the moment about whether there will be or will not be, there may be, a mortality signal with Remdesivir. But that is a bit different to dexamethasone. It's an unlicensed product in the European area until licensed by the EMA. And it is a medicine that is, as I understand it, relatively difficult to manufacture, and because it is not yet licensed, will be in much less plentiful quantities or less easy to obtain than dexamethasone.
I understand. And will you make a separate assessment of it, or will you draw on the US trial data or the European assessment?
Well, in a sense, the assessment has already been made, in the sense that the company concerned have applied for an emergency access to medicines scheme run by the MHRA. That has been granted, and already patients in the UK healthcare system, the NHS, can obtain Remdesivir.
And
there is a protocol for that. It has been available now for several weeks, right, and stocks are available.
So it's open to any NHS physician treating people with COVID to prescribe it?
Subject to certain conditions and in hospitalised patients, yes.
Thank you. And finally, you'll have read today that the US has announced that it's bought up the prospective production, I think, of Remdesivir, 100% of the production for July, 90% for August and September. And Germany, I think, has said that it has secured enough supplies. Do you have a concern that it's not going to be available to UK patients?
So right now there are stocks in the UK, and I judge us to have adequate stocks of pre-licensed, as in unlicensed clinical trial stocks of Remdesivir, which are available to be used through the emergency access protocol that I talked about. But it illustrates the point that depending upon which medicine you discover works for COVID-19, if it's one of the ones that is already licensed for something else that one can repurpose, then it tends to be plentifully available. And if it's an old drug, there are many manufacturers often making generic versions of the same drug. But if it's something brand spanking new, as it were, from a new developer, it is likely to be in relatively short supply in the first instance. But I do know that the commercial teams in the Department of Health and Social Care are working extremely hard in this area.
And is it a function of the Taskforce to secure the supplies of therapeutic treatments for the UK that are manufactured overseas like this?
Yes, so the Taskforce concerns itself really with the end-to-end journey from identification of medicines that require evaluation in case they are effective in the treatment of patients with COVID-19, through to colleagues within the task force, within the DHSC commercial teams, who will take responsibility for acquiring those medicines for the UK. And I'm sure you'll understand it's a very fine balance between Professor Sir so-and-so saying this is a very important drug that you need to evaluate, through to full proof of concept through a large trial such as Recovery. And it is a finely balanced judgment about when or if, well, first of all, if, and secondly, when to obtain those medicines in quantity that may be needed for patients. Yes. But unlike a vaccine, we're never talking about needing 50 million vaccination courses for the whole of the UK population. Each of these medicines will have potentially a point in the course of the illness when it's most beneficial to give it, a point in the course of the illness when it becomes futile to give it, and there may well be medicines that work in certain subgroups but not in others. And typically that's going to be divided into the antiviral drugs such as remdesivir, where we know with antiviral medicines, because of the nature of the way that viruses multiply in the body, that the earlier the treatment, the likelihood is you're going to get a better outcome. But we know with COVID-19 that you have this vicious tail end in the illness, which is essentially a hyperimmune response, where damping down that immune response is the most important thing. And that's where dexamethasone comes in and plays its role. So there's almost kind of different horses for different courses along the course of the patient pathway.
But for that early treatment, if it proves to be the case that remdesivir turns out to be a useful therapy, do you believe that you can obtain it in the quantities that we would need in this country, given what the US has done in terms of buying up the stock or the future production?
Yeah, okay. So I think the answer to that lies in two parts. No, I don't think it will be as easy or as straightforward to obtain as the measures we took for dexamethasone by virtue of its scarcity and by virtue of the fact it's a new medicine with a relatively long manufacturing time. So no, I don't think it's going to be as easy. I couldn't say otherwise. Okay. By the same token, it won't be suitable for all patients. It's a intravenous medicine given through a vein. Until there's an oral formulation, it would never be suitable for use as early as we might like to give it, for example, in primary care or in patients at least at home with it. That's just not a reality in any healthcare system in the world for a medicine that's given through a vein. So there will always be these limitations according to the formulation that the company has developed so far.
Thank you. That's very clear. Graham Street.
And I'd like to ask you some questions on clinical trials, but before I do, can I take you back to the point you made in response to the previous panel, that you said we were as well prepared, if not better prepared, than any country in the world for a flu epidemic, and we used that experience. If that is the case, why are our statistics on infection rates and deaths, why do they look so bad in comparison to other countries?
Okay, right. So this is not flu. this is a coronavirus. The point I was trying to make, I think, well, I'm sure I was that, for example, listening to the previous discussion, we had to start from scratch, or nearly from scratch, to make a coronavirus vaccine. And I'm part of the Vaccine Taskforce. You've seen the amazing work that's happening there. But were that to have been pandemic influenza, for example, the UK has, still has, and has had for many years, an advanced purchase agreement with vaccine manufacturers who make flu vaccines to have very, very rapid access indeed to a pandemic flu vaccine, as fast as any other country on the planet. And the advantage there, of course, is that we already make seasonal flu vaccines, and effectively what you do is you change the formulation and turn your seasonal flu production capacity towards making a pandemic flu vaccine. So the point I'm making is that actually we are very well prepared indeed for a flu pandemic, but we have, the whole world has been very unfortunate that this has been a coronavirus pandemic instead.
How effective has the process for awarding clinical trials national priority been, and how much has it speeded things up?
So there was a very, very rapid response. I'm just going to look for the precise timelines for you. There was a very rapid response to funding clinical trials in the UK. This is really down to the hard work of the NIHR and the UKRI together, who launched a research call for COVID-19 on the 4th of February. That, I think you'll agree, is very, very early indeed. That was originally badged to be a £20 million research fund, but grew to 24.6. The first applications closed on the 13th of February, so you can see the time frames are very compressed. But we already had 58 applications in that space, and the second part of the call closed on the 27th of February, and there were 212 further applications in that space. There was a rapid response assessment panel that met to consider the first bids on the 2nd of March, and by the 23rd of March there'd been another meeting and the first 6 projects had been formally announced. And what we were looking for in those projects, and Dr. Sheffield will be able to give you more detail again, were research projects that could lead to changing or influencing the UK's response within 12 months. In other words, we were looking for very rapid impact studies. That wasn't the only focus, but the majority focus was what is going to get us something useful quickly. And since the 17th of April, there has been a rolling call, which closed yesterday, in fact, and has been ongoing ever since in that space, funding to a much larger amount than the initial 24.6 million. So I think that tells you two things. One, that the UKRI NIHR research system, the government funded research system, responded incredibly quickly. 4th of February is very early to put out a call if you think about the timelines of what we knew about China and how it was happening. And the response from UK researchers has also been overwhelmingly large and swift.
This was all done under the European Clinical Directive, was it?
I beg your pardon, I didn't catch that.
Sorry, I probably didn't. Were the clinical trials carried out under the European Clinical Trials Directive?
All clinical trials are carried out under existing EU clinical trials directives, yes.
Right, thanks. Has it been difficult getting patient participation in the clinical trials, and what has been done to increase that participation?
Right, well, so I've got an amazing story to tell you really on that one. The recovery trial, since it started, has recruited perhaps just shy of 12,000 patients. That's really the most unprecedented and astonishing speed. As of the 3rd of June, the Solidarity trial, which is relatively similar, run by the World Health Organization across multiple countries across the globe, 35 countries together have recruited 3,500 patients. One country, the UK, has recruited almost 12,000 patients. Almost 200 UK hospital centres have taken part in that amazing effort. So, yeah, I think the world is looking at us and saying the UK clearly can mobilise its physicians through a nationally government-funded research platform, and those physicians in turn can mobilise the altruism of UK patients to take part and help the world get answers.
That's extraordinarily impressive. Apart from people's altruism, is there any procedural, organisational lessons to be learnt and recorded from that process?
Yes, and I think at this point you should really, with respect, be asking Dr. Sheffield to elucidate the might and the organisation of the NIHR and the clinical research networks. But I think I would pay particular tribute to the 4 UK chief medical officers who responded very quickly when we asked them for a national letter to clinicians that would really spell it out to everybody working on the front line that this was a national effort and that it was very important that as many patients as possible were recruited into the recovery trial, which at its peak, when unfortunately we had a lot of COVID-19 patients, was recruiting well over 200 patients per day across the UK. And if you kind of compare that, for example, to the size of the remdesivir pivotal studies of under 1,000 patients, we were recruiting at over 200 a day.
Thank you. Dr. Sheffield?
Yes, so I'd like to sort of reiterate that the massive effort that went in across the whole of the UK to deliver these trials. Just to give you some more close facts on recovery, we set up 176 sites, so 176 hospitals across the country started to deliver the recovery study. It was given an urgent public health badge on the 11th of March following its funding. The HRA, Health Research Authority, approved it 4 days later. The Medicines Health Research Authority also approved it in 4 days. So there was amazing synergy between the Medicines Health Regulatory Authority and the Health Regulatory Authority. And the trial opened on the 18th of March and the first patient was recruited on the 19th of March. And in actual fact, at its peak, during April and May, over 400 patients a day were being recruited. And what I would like to say is that this was an effort carried out by all the research networks across the NHS, and NHS staff themselves were highly committed to this. But most of all, patients actually volunteered to be involved in this research in their most desperate hours. And if you look at the results from the recovery trial, many patients didn't survive that, but they still committed themselves to be involved in research. And in fact, in total, over 120,000 patients have been recruited into clinical studies across the whole of this epidemic. That is a massive achievement by the UK.
Thank you. And final question from me. At the start of June, there were 720 proposals for research funding. How many are there at the present time? And do you have a view on how many of those are likely lead to positive therapeutic processes for COVID-19? Sorry, that was a clumsy question, but I think you
Who's that to?
I think to Dr. Sheffield, but...
Dr. Sheffield?
Yes, so I'm not an expert on the actual grant aid side of the NIHR, but we have had an enormous number of research grant applications. We've prioritised those that were already funded, because that would make it a lot easier for us to be able to deliver them. We are still going through a process of sifting and sorting through these studies. The studies are highly varied in the quality that they come to and in their state of preparedness. The whole point about our urgent public health process was to make sure that those studies were ready to go so that we could get them delivered quickly, efficiently and effectively. Our aim was to make sure that these studies were completed within the year, which is exceptional again in the terms of the way that we deliver research.
Thank you.
Thank you. Aaron Bell.
Thank you, Chair. I'll stick with Dr. Sheffield for now. We've already heard about the achievements and the successes that we've seen, especially dexamethasone here, but also what's going on with remdesivir. There have been a couple of controversies along the way, obviously, and Professor Van-Tam's already mentioned hydroxychloroquine. How did we handle that here? What lessons have we learned about what happened with that and around data integrity and peer review processes? Was it a major issue for us, or essentially is it something that played out in the academic sphere rather than your area, Dr. Sheffield?
No, it absolutely played out in my area, because the issue that we had with clinical trials was actually about global supply of drugs. Even simple drugs like hydroxychloroquine were actually very difficult to source for the clinical trials. And it was only the fact that we had a system with the Department of Health procurement systems that we actually had those supplies available, working with Public Health England, for us to actually conduct those trials. What was really interesting from our point of view was that a lot of countries, I would say, panicked before the science evidence was available. So we know that there was permission given in France to prescribe hydroxychloroquine with no scientific evidence of effect. The FDA also allowed hydroxychloroquine to be prescribed. So again, with no scientific evidence. So what was remarkable was the fact that the chief medical officers really made that statement very early on in the epidemic, stating quite clearly that there wasn't any scientific evidence of appropriate treatments for COVID, and that the doctors and nurses, those people caring for people with COVID, needed to be involved in research. Research was going to provide the answers. So when we had all the issues with hydroxychloroquine and the safety issues, and our trials were suspended, it was the fact that we had recruited a large number of patients already into the recovery trial with hydroxychloroquine that we were able to submit that evidence to the MHRA to demonstrate quite clearly, and that was, again, the recovery team doing that, but quite clearly that there weren't the same level of safety issues in the context of our clinical trial that they were seeing in the paper that was published in The Lancet and subsequently withdrawn. And I think that was a useful exercise to demonstrate why the science base was so important. Secondly, because of the fact that the recovery trial continued, we were able to, a fortnight later, publish evidence that demonstrated that actually hydroxychloroquine had no effect on mortality on patients that were hospitalised. So in actual fact, we drew a close on that piece of research, and to me that was as important a discovery as it was the fact that dexamethasone worked. There'd been a lot of hype about hydroxychloroquine and its effect, and what we have demonstrated that in hospitalised patients with COVID, hydroxychloroquine does not work.
Thank you. You're absolutely right, of course, that negative results are equally valuable in terms of moving us forward and determining what the truth is. I was struck by your use of the word panic there amongst the international response, because I think that speaks to what happened with the lockdown in the first place, that the countries who panicked locked down earlier, and then that seems to have played into their favour. So when dealing with a pandemic, is there not a case for sometimes taking a more precipitate approach, which we call it, rather than necessarily waiting to play it out like you normally would do? Is that something you consider?
I mean, as an ex-medical director of a large teaching hospital, I believe that the way that we practice medicine has to be on an evidence base. So I believe very clearly that there's a tripartite agenda in healthcare. Firstly, that we deliver the highest quality clinical care. Secondly, that we make sure that the education and training of the staff is of the highest level. And thirdly, that we continue to deliver research that gives us the actual definitive answers as to how we should treat people with their diseases. So I'd be very uncomfortable if we move to an area where we listen to what was going out on Twitter feeds, on a whole range of fake news about products that may have an effect. The fact is that we have to base our medical care on the best scientific evidence available at the time.
I completely agree with that. It's more a question of whether we have the stocks of something that might prove successful. Could I just also ask you briefly about ibuprofen? Have you done any clinical trials around that? Because there was controversy over whether that was contraindicated earlier in the term, or I know that's been resolved to some extent, but have there been trials about the effect of ibuprofen and other anti-inflammatories?
There is a trial being conducted at the moment on a liquid format of ibuprofen. It started late because of the controversy, so we haven't got the evidence based on that trial. It didn't receive urgent public health badging because it wasn't allowed to start until towards the end of the first wave of the pandemic.
Okay, that's great. Thank you very much, Chair.
Thank you very much indeed. Can I come to Katherine Fletcher, please?
Thank you, Chair. I just want to understand what else could be coming. But before I do that, can I just add my voice to the record of what an amazing response this has been? I mean, genuinely, that's remarkable. And it's testimony to the British public being marvellous, as well as our structures and scientists. Is there anything else coming? We've done a remarkable thing. Is the indication that dexamethasone is something that works, sending people off to look at other drugs that interfere with the immune system? What are your thoughts? I can see Professor Van-Tam waving his learned finger at me. So,
Professor Van-Tam.
Thank you for the question, and obviously I think that's a very pertinent question for you to ask. By way of a bit of context, before I answer your question, I will say that now we are pretty sure that dexamethasone is pretty important, or at least shall we say a corticosteroid is pretty important in patients who are admitted to hospital who require oxygen. They're at that stage of severity. Right. The new standard of care is therefore dexamethasone or an alternative. And from that perspective, the comparison now becomes what else can you add where dexamethasone is part of the supportive care or standard care arm. So the question has now altered. It's what is the incremental benefit of adding something on top of
so you don't mean like adding prednisolone on top of Dex?
No,
you mean the complementary?
Yeah.
Okay.
I mean, Dex is a constant now.
Yeah.
As the underpinning therapy. So it's kind of what else do you get back? Now, recovery has not finished. It isn't finished by a long chalk. And if, regrettably, as Professor Bell said in the previous session, there is a resurgence of disease this autumn, winter, recovery will again be busy and continuing to do its work. I am expecting recovery to be able to give us a readout fairly soon on another of its arms, which is azithromycin. Now, I recognise that that is an antibiotic, and the listeners, not the committee, but the listeners will be saying, well, that sounds very bizarre for a virus. Azithromycin, as you know, has a very potent anti-inflammatory effect in the lung, and that is the reason why it was chosen as a potential arm. And on top of that, of course, it is widely and easily available, and that's also important to us, because we can discover anything wonderful we like, but if we can't actually get it and give it to NHS patients, what does it mean? So from that perspective, I expect azithromycin, the recovery trialists, to be able to give us some pronouncement on that fairly soon. And they are also evaluating an IL-6 inhibitor called tocilizumab, which
Sorry to interrupt you. Forgive me for being nerdy. We probably should explain what an interleukin is for the benefit of the take.
In other words, it's a sophisticated drug that damps down the immune response in those very severely ill patients. And again, the phenomenal success of recovery is that there are many hundreds of patients already randomised to that treatment, and I think it is likely to be, when it reports, and it's not ready to report yet, the largest study of an IL-6 inhibitor in the world, and therefore most likely to give the most solid signal. That's not the end of the story. Recovery is also now evaluating convalescent plasma, which is the plasma from patients who have had proven COVID-19, have recovered, and therefore have antibodies in their blood. The plasma will contain the antibodies. That can be infused back into other patients who are notably unwell with COVID-19 and may be of benefit. There's certainly quite a historical literature that convalescent plasma may be important and may reduce mortality in severe respiratory virus infections, although obviously not proven yet with COVID-19. And that's another readout that I would expect from recovery in due course. But beyond that, the Therapeutics Taskforce has a committee that is looking deeper and further afield for novel medicines, either repurposed or completely new, which might have a role to play in the treatment of COVID-19, even though they were not initially intended or designed for that. And those trials have started in very small scale. We plan to try and incorporate more of them into recovery, to use that kind of national branding and national footprint to get them going a bit more. That is going to require the patient numbers. And of course, at the moment, disease activity is thankfully low in almost all parts of the UK. Just to give you a flavour and to give the committee a flavour of the kind of things that are going to be looked at there. There's a novel antiviral called bencentinib. We can send on this list to the committee.
Thank you. It'd be very great.
There are a range of different monoclonal antibodies. There's some tyrosine kinase inhibitors, a protease inhibitor. There's a common drug, spironolactone, that reduces ACE2 receptor expression in the lung and therefore may be important in preventing the virus from binding in the lung. There are some anticoagulants in there, both parenteral and inhaled or nebulized. So there's quite a lot there, trying to target, and without going into the detail, multiple different pathways in the pathogenesis of COVID-19. And that's one of the pieces that I would say that we don't fully understand those pathogenic pathways yet. That would be tough going for a virus that we only first knew about 5-6 months ago. So from that perspective, that's a voyage of discovery too.
Thank you.
That's
Katherine?
Forgive us, Chair. So I think what we're worried about is a second spike coming in winter as, you know, people start spending more time indoors. And the wonderful efforts that have gone in to stop the NHS getting overwhelmed by very sick people. So lots of what you're talking about there is about reduction in mortality and helping people spend less time in hospital. Is there any indicator or any tests that are going on to stop disease progressing from the point where you need to get admitted to hospital? You know, almost minimise its, I've forgotten the proper word, but, you know, minimise how awful it is. Maybe Dr. Sheffield, could you help?
Yes, so we have run also the principal trial, and that was incredibly fraught with difficulties, mainly because it was aimed in primary care with that specific purpose of preventing patients or helping, hopefully stopping patients from getting to the state where they're admitted into hospital. We had enormous problems delivering that trial because of the really the lockdown of general practice. So we had to continually adapt the design of the trial and also how we got the supplies to people that were in lockdown. There was a whole chain of really complex logistical problems in delivering that trial. So what we've done over the last 2 months is really try to redesign that trial in a way that it will be much more effective if there is a second wave, because we're going to have to expect that patients will only be seen remotely by primary care. We set up a call centre with the NHS 111 system, so that when patients were reporting their symptoms, which was more common than reporting them through that route than through their own primary care practices, that we could then put them in touch with the central primary care research team in order that they could be recruited into the study. And what we're looking at the moment is what products would we be interested in aiming at that group of people. So in fact,
and where are you in that process, just in terms of understanding what we could be evaluating?
So well, one of the things that we're looking at the moment is convalescent plasma. So we already have the study going on in recovery and in REMAP-CAP, our ITU-based platform study. But what at the moment is being written up by National Health Service Blood Transfusion Service is a model for taking that convalescent plasma study out into the community when people are infected early. Now, I think we feel that might have a much bigger and more profound effect because you're treating the virus at an earlier stage. But the logistical problems of delivering a trial in those circumstances, being clearly identifying that the people have got COVID and the testing, is something that we'll really have to work hard at to make sure that we've got all the systems and processes in place in preparation for that.
Never mind getting the volume of convalescent plasma available.
Well, great progress has been made on that. So that was the initial problem. So when we started the convalescent plasma trials back in May, supply was the problem, because you have to wait 28 days for the convalescent plasma to have the levels of antibody. And what we were finding that it's the sickest patients that produce the most antibodies, and at 20 days post-infection, even if they've recovered, they're still very ill. So 40% of the people that were donating their plasma weren't fit to donate their plasma. So we've had to make all sorts of adjustments, and the National Health Service Blood Transfusion Service will be able to explain that in much more detail than I can. But there are over 9,000 units at the moment in storage, and that collection is continuing. And the advantage of the convalescent plasma is it's a local resource, and that it also it can store for up to 3 years. So we are building stocks of that at the moment, and the really interesting question will be, does it work in recovery? Also, does it work in early disease in the community?
Thank you, Katherine.
One more for me. Yeah, that's really A, scientifically interesting, and B, good luck. I'd love to see the results. What that's not going to be is something that's made available to everybody that hasn't had COVID by a volume. Is there any? I mean, it doesn't matter if the answer is no, but is there any indication at all of a test where we can all pop a pill if we get it, and that helps us minimise the worst side effects?
I think you're illustrating a tension here that the lower you go, if you like, in the patient pyramid, the much greater numbers you have in primary care. And so anything that you're going to discover that works at that early stage that can be given to patients whilst they are still a bit poorly, but not very poorly, in the community. First of all, you've got to be able to deliver at scale. Real scale, which means very high volumes of supply. And on top of that, it's got to be safe enough that it can be administered in a relatively unsupervised community setting, which is very different to the 24/7 staffing on even a general hospital ward. And so from that perspective, it's a different challenge altogether. And right now, even if you were to discover something that was highly safe but completely new, and orally administered, pop a pill, as you say, just once, fire and forget, then to coin a military analogy, fire and forget. But, you know, from that perspective, if it was a new medicine, you would have to be secure on supply as well and manufacture. And that's another headache that comes with this whole area.
Yeah, we're a long way off. Thank you, Tom.
Thank you, Katherine. And finally, for me, just on that point of manufacturing, you might have heard in the previous session we were talking about vaccine manufacturing and the establishment of the vaccine manufacturing centre. Is your task force thinking in similar terms about the need for a therapeutics manufacturing centre, or does the vaccine centre, is it able to cover both, as it were?
Right. So the final question is it able to cover both? No. I'm the former UK medical director of a vaccines company many years ago, and the manufacture of vaccines versus the manufacture of medicines is really very different indeed, and the manufacture of medicines is very, very diverse indeed, from, you know, the typical kind of dry active powder that can be compressed into a pill and placed into a foil sheet, through to many of the medicines I talked about that we want to evaluate next, which are complex monoclonal antibodies that really have, for a start, they are proprietary and patented. For another thing, they are incredibly complex and difficult to manufacture. And I think trying to find a generic solution to drug manufacture is very different indeed, to finding a generic solution for vaccine manufacture. Even that's not straightforward. But for example, if you have a chemical or a cell culture bioreactor, if you've got both of those, you can, in theory, make a lot of vaccine. The different chemical processes involved in all these very diverse medicines is really so difficult, and so you can't just kind of have, you know, one or two industrial options that one size or two sizes fits all. That is not to say that the UK would not be perfectly prepared to look at manufacture of a medicine that could be manufactured if it had to, but really it's not in the same space where you can invest in the facilities and then wait for which vaccine makes it, if you like. This is a different space where you have to know what's going to make it, and then say what's going to make it through the clinical trials process, and then say, now how do we manufacture it? And can we even do so without, you know, in terms of its patent status?
Indeed, and obviously we've talked extensively about the benefits in taking therapies that are familiar and have been tested and have been used for other things. But if there are some therapies that are new and require manufacturing facilities to be built up, is the UK a contender for that, and is there anything we can do to make ourselves so?
I don't really have the answer to that question personally.
So it's not.
So I wouldn't want to just speculate and give you some kind of. I won't answer.
And so for the task force, obviously the vaccines task force very much has this in scope.
Yes.
That's not something that your task force. It's not part of the remit of your task force. Is that right?
The therapeutics task force very much has that within its scope and within its remit. It is currently reporting in through BAYS, so it's reporting in through the right piece of government to have that ambition for manufacture. But what I'm saying to you is that right now there is nothing obvious in that target space where we need to be working at the same pace or intensity that we have had to work from day one in the vaccine space.
I understand. And then perhaps conversely, there may be a need for commercial negotiators to negotiate supplies from other manufacturers around the world for therapies that might be licensed elsewhere.
Yes.
Is that right?
Yes, indeed. Yes, absolutely.
Thank you. Well, that's understood.
Thank you very much, gentlemen.
We have a division that explains the bells. We'll Just perhaps a final question, if you can still hear me, Professor Van-Tam. Can you above the din? Good.
Yep.
We talked a bit with Professor Bell, Sir John, about what we've discovered through the pandemic. We know much more now than we did at the beginning, and one of the things is that we know that asymptomatic transmission is a pretty common feature of most countries in the world, and therefore practice should develop in response to that. But what are your reflections on the implications, perhaps as Deputy CMO, of the testing regime in NHS settings, given that we know that upwards of 42% of people may be asymptomatic?
Yes, as I understand it, and having checked recently with NHS colleagues, there's now plenty of capacity for testing, and every single patient that is admitted either for elective surgery or as an emergency admission, whether that's for Covid-19 suspicious symptoms or for something else like a broken leg, is tested for Covid-19. I think that's really important. I think the programme of testing and having a low threshold for testing in staff is important, but I do think there are limitations to what you get back from testing. And let me be clear on this, that the incubation period for Covid-19 may be typically 5 days, but is described up to 14 days. Now, if you were an individual, sir, who was infected now, and you are ordained by a higher power that your incubation period is going to be 10 days, and you were infected yesterday, I can test you on every day for the next 8 days, and your test will be negative. I will only hit the bullseye, if you like, on day 8, 2 days before your symptoms begin. And so you can see the problem you have in terms of false reassurance of negative tests, unless you are testing extremely frequently indeed. And you have to think very carefully about whether 2.5 million workers in our health and care sectors, how you could do that in an extremely frequent way right across the piece. So that's just a word of caution that yes, testing's incredibly important. Yes, we are now in a place where we can do a great deal of it, but there are limitations to what you can get back in terms of reassurance or management tactics from a negative test.
I understand that, but that applies for every negative test that test results from anyone.
And indeed at the moment, because we have a very low level of disease, you will get false positives too.
Indeed.
And so this is not magic science, if you like.
In general, we now have enough testing capacity to be able to have the option of testing people. Is that your assessment?
My understanding is we have a lot of testing capacity now, and that may well be important if we get a resurgence of disease in the autumn. And it's very important for our test and trace facility going forwards, and it's very important for testing all our patients who are admitted to hospital for other reasons.
Yeah, indeed. And just a final point, in terms of the false reassurance of a negative result that may be before the disease is detectable, is it not the case that if you don't have tests at all, then in effect you're drawing that same false assurance through not having symptoms? And if you continue to work in a setting in which you're with vulnerable people, are you not at least giving some more useful information?
Yes.
You're closing down
Yes.
the level of
I'm absolutely not arguing for no testing. Absolutely not. I'm just making, I hope, a very clear point that testing is extremely important, but it is not without its limitations.
Indeed, that's very clear. We're very grateful to you both, not just for your evidence today, but as my colleagues have said, for your extremely important and extraordinary work. We're very grateful for everything you're doing on behalf of the country and indeed the rest of the world when it comes to some of these discoveries. And very grateful for you giving evidence to us today. We want to make sure that we learn the lessons so that we can make sure that we apply them to do all the things that we've done well during this pandemic deliberately in future, so that we can build on the successes, as well as if there are learnings to be made as to how to do things differently, we can do those together. So that is our purpose, and we're very grateful for your assistance with that endeavour today. Order, order.